If not all of you know already our speaker of today, Alex Brogan. He obtained his master and PhD from the Department of Chemistry, University of Bristol. After a short postdoc there, he moved to the Department of Chemical Engineering, Imperial College London, when he was a research associate until 2019. During this time, he was also a visitor researcher at the Koch Institute for Integrative Cancer Research, Massachusetts Institute of Technology. In 2019, Alex moved to the Department of Chemistry, King's College London, to establish his group as a lecturer in chemistry. His research is broadly on the development of protein-based biomaterials with high stabilities against temperature aggregation and not aqueous environment. Particularly, his research focuses on combining these biomaterials with ionic liquids to provide new opportunities for sustainable biocatalysis and soft material design. Thank you, Alex, for giving a seminar today. Maybe I will say something about the Net Zero Center after, but I would like to have your presentation start.
Okay,
thank you.
Yeah, excellent. Well, thank you for the invitation to talk, and thank you everyone for turning up and finding this room. King's Building isn't the easiest to navigate. So yeah, as introduced, I'm Dr Alex Brogan from the Department of Chemistry, and I'm going to be here to talk to you about some of the research that we're doing, particularly with a focus on the sustainable biocatalysis for sustainable chemical production. And I thought I'd start by justifying why I feel this is net zero. And basically, part of this is net zero isn't just a commitment to reduce carbon emissions; it's a commitment to ensuring both the sort of sustainability and resilience of the economy in totality. And so we're going to think beyond simply reducing those carbon emissions from typical things like electricity and ground transport to start thinking about how we can build in resilience to production, circularity into production, and generally decarbonising. And so thinking of moving away, particularly for chemicals, etc., to more sustainable resources. Which then brings me on to what we're after. So, the research that we do predominantly in this area is thinking about how can we go from waste, biomass, and other renewable resources to form the fuels, chemicals, and plastics that we require for modern life. And it may come as no surprise that I feel the answer to this is in biology, and so harnessing the toolkit that biologists provide us, so enzymes, to produce these things from these things in a sustainable manner. And this allows us to not only build in a production system that might be more efficient, but also we can start thinking of circularity in terms of the resources that we use. So we have enzymes that can catalyze a vast array of reactions, and actually, to date, any reaction that you put your mind to, you'll eventually find an enzyme that will do the job. And especially now with computational techniques to design in new enzymes, this is getting easier and easier. The problem is now, though, is that the fuels, chemicals, and plastics that we require for modern life require solvents that biology is not amenable to. So these will require non-aqueous environments to make in any sense of the word. And if we think about moving away from a petrochemical system to a biorenewable system, the easiest way of doing that without any expenditure on new facilities is to do what we would call a drop-in sort of scenario, where we try and make things as convenient for the swapping out, basically, as possible. And so the biology is not compatible, effectively, with those typical environments required for this production. So they are often biocatalyst enzymes. Biocatalysts are often insoluble and inactive under non-aqueous systems, so organic solvents. Well, this, for me, as introduced, is as a biomaterials chemist, I feel that this is a biomaterials problem. So we have two key biomaterials in my lab to look at the solvent-predicted proteins and ionogels, and today we're going to focus on solvent-predicted proteins, particularly as a platform technology for stabilizing biocatalysts from temperature and aggregation, and actually non-aqueous solvent as well. So these solvent-predicted proteins, what are they? They are a modification strategy to protect enzymes from various things. So I'll go through this briefly. That this is, we take our enzyme, which is which has a heterogeneous charge, so it's both negative and positive on the surface. We perform some very straightforward catalyzation processes, so this is EDC-mediated coupling. This homogenizes the surface charge of our protein to yield a cationic enzyme. This then allows us to electrostatically couple anionic surfactants to form what we refer often to as the nanoconjugate, and this introduces now the protection there. So this is a biomaterial strategy which basically introduces a canopy around the surface of the protein that protects it from a whole host of aspects like temperature and solvent. We can actually dehydrate this and anneal it to form a fluid which is devoid of any water, which then allows us finally to introduce a dry, stabilized enzyme into non-aqueous solvents. And as I'll touch on later, the solvents we've chosen are a sustainable class of solvent called ionic liquids. And so here we can just see that just comparing, there's a whole range of polarities for ionic liquids. There's all the way from hydrophilic to hydrophobic. And here this is unmodified protein, this is modified protein, so I'll often refer to this as modified protein as well. And we see then for the unmodified protein, the hydrophilic ionic liquids, the enzyme will go in, kind of it's a heterogeneous sort of mixture, but once we've modified it, it goes in and it's a homogeneous mixture of enzyme and a sustainable solvent. Similarly, for the hydrophobic solvents, this is the unmodified enzyme. In this case, the protein does not go in at all, but once we've modified it, we see that the enzyme will go in and is a homogeneous mixture of enzyme and ionic liquid, which then becomes very useful when we start thinking about using these for, in this case, biocatalysis purposes.
I mean, the thing is, of course, the protein reacts via some surface sites. Do you hide these surface sites as well? I mean, so does it keep its functionality as an enzyme?
Yeah, and that's the whole part of our research is maintaining that. So a lot of my students have done a lot of good work on actually enhancing sometimes, so particularly the more surface sort of accessible active sites. So for example, our lipases, a lot of them are cleft on the surface, and what we're actually seeing is under multiplication, come back to this, is that we're seeing an enhancement. So anyway, the material that we're looking at, I'll show some data, and you don't have to take too much away from it. I'll explain what it's showing. But this is circular diachromosome, so we're very fond of this in the lab. This tells us about the structure of the enzyme. So the enzyme structure is intimately linked to its function. So if we understand the structure, we will have a prediction of the function. Long story short is we can see that for this particular protein, it was maintaining its structure, but over a significantly increased temperature range. So we're seeing that the protein is stable up to about 160 degrees Celsius, and this means that we've expanded the window from where it's stable under aqueous conditions up to about 70 degrees Celsius. We're now increasing that to 160 degrees Celsius. I won't go into details too much, but we did some fancy neutron scattering experiments to show that the dynamics of the protein, after we've modified it, are the same as in the absence of water as it was in water. This tells us that the protein not only has structure, but also has dynamism, and this is very key and important to the enzyme activity, which we can show here for. This is just the material, so in the absence of any water, we're able to show that enzyme activity is not only maintained, but we can activate it up to 150 degrees Celsius. And then I'll just skip to just say that we did some nice experiments on a protein called lysozyme to show that this is through the unfolding pathway, we were able to determine that a otherwise reactive intermediate on an aggregation pathway was shielded from aggregating in this biomaterial. And this is all derived from that canopy. And so we're introducing a surface modification for biological entities that protects it from temperature and aggregation, but whilst maintaining the dynamism and increasing the thermal stability as a result. So this is what we term the solvent-free proteins, and you might think that we want to use these directly for various purposes, but the problem here is that these have very high viscosity, and so its viscosity is similar to cold treacle, which isn't very useful for processing. And so we turn to ionic liquids as designer solvents, but with a quite sustainable action to them. And so for those of you who don't know, they are effectively salts, they're organic salts. with highly tunable properties, so there are a near infinite number of combinations of cations and anions that will form liquids at room temperature. So for us, a very straightforward system is to use this very common cation. This is often off-the-shelf ionic liquids. This is the ethyl-methyl-methazolium cation. And if we swap out simply for these four anions, we get liquids with very similar physical properties, but the chemical properties are very different. And so we have, on this side we have very polar ionic liquids, which are able to solubilize a vast array of various substrates, up to hydrophobic ionic liquids, where the solubilization is actually very more selective. And so we can actually tune this, in theory, to be selective for whatever we would want, which is great. And so in particularly that we're interested in for us is that the fact that these ionic liquids are much better solvents for polymers and otherwise recalcitrant substrates we can't get into, those of us in our liquid field, called the molecular solvents like water and other organic solvents. So this is key for us, and actually there are other properties that we're currently exploring. Thermal stability is important, so and so is the negligible vapor pressure. This allows us to operate reactions at higher temperatures rather than for a pressure, so we don't worry about the solvent boiling off. This in fact actually means that these solvents are very recyclable, so we can reuse these solvents, and there's some excellent case studies, not from our group but elsewhere, on reusing these near infinity. But so far, the recycling, I think, one company, a pilot company, has been recycling the same 200 kilograms of iron liquid for about 5 years. So they've been using that process to be cycled over and over and over again. Something that we're looking into, but I won't touch on today, is looking at the broad electrochemical window. This also provides opportunities for biocatalysis, and I won't touch on today. So this all started thinking about how can we utilize those sort of aspects of the renewable resources, so thinking of biomass to start off with. And so initially we're interested in the conversion of cellulose to sugars, which is the bedrock for the second generation biorefining. So can we provide a renewable resource, so cellulose from lignocellulosic biomass, for the production of sugars and other platform chemicals. And so the first step in this is really treating the cellulose with a trio of enzymes, and these are the cellulase enzymes, where we have our first enzymes. We take crystalline cellulose in aqueous conditions. The endocellulose comes in and it basically hydrolyzes and it sort of solvates aspects of the crystalline cellulose, such that the second enzyme, the exocellulase, can come in and make soluble fragments of the cellulose, which the final enzyme, glucosidase, will come and break down into the sugars. Now, this is the mechanism that fungi use when they're growing in the woods, and you can imagine that is a very fast process. And so the rate limiting step for that is actually this step. So this enzyme is the bottleneck in the formation. This is actually a defensive tactic for fungi when make sure that bacteria don't outcompete them. Probably too much information, but it's interesting what it's saying. Anyway, so we were wondering then, can we use ionic liquids then as a better solvent for saccharides to speed up this step? So we were thinking, what if we introduce this enzyme, ignore this step, but can we use the other enzymes in consort by improving access to the cellulose fibers, because we know that better solvents and polymers? Would this speed up this? Initially, we were interested in this bit because this is as I said, it's the bottleneck. So if you buy this cocktail of enzymes from Novozymes, the ratio of enzymes is typically 1 to 1 to 90 to overcome that rate limitation. So we're thinking maybe we can just improve that. Long story short, we modify the enzyme, we get it into ionic liquids, we assess the structure against ionic liquids. So again, this is looking across the range of polarities. Long story short, this tells us about the structure, and by and large, it is maintained. So we've maintained the structure of our enzyme once it's been solubilized in ionic liquids, which is fantastic. We did some thermal stability measurements to show that we've also improved the thermal stability of this enzyme. So in this case, whilst in water, I had a maturation temperature that tells us about the stability, so the maximum temperature when you can use this enzyme at, of 71 degrees in water. In this acetate ionic liquid, something that is very interacting, it will disrupt hydrogen bonding. We saw that stability was increased to 117 degrees Celsius. And then in the hydrophilic and then hydrophobic ionic liquids, we increased as well as nearly 140 degrees degrees Celsius. So we were able to solubilize the enzyme into ionic liquids and they become highly stable. So we looked then at the enzyme activity. So looking at, here is the unmodified enzyme, and this is the modified enzyme. So in aqueous conditions, we see that there was no change in enzyme activity. So we were able to modify it, but we saw very little change in enzyme activity as a function of that modification. And when we move to the ionic liquids, so this is a different ionic liquid for these ones, but this is after screening. This was the best one given a whole bunch of parameters. And we see the enzyme activity drops for the unmodified, but it increases for the modified. And actually, this is a solubility issue. So when the enzyme is less soluble, it's less dynamic, it becomes a bit more of a powder, it's less able to do its job, and so that's why I see this enzyme drop. And then because we've increased the solubility and we've made it a better solvent for our substrate, we see this slight increase in enzyme activity towards that soluble fragment of cellulose. And actually, well, we then start thinking about, well, this is an enzyme. These are ionic liquids are good solvents for polymers. What about if just put a polymer into ionic liquids, do we get a reaction? The answer is yes. So we see here then, this is a reaction that isn't observed in water, but we see it in ionic liquids, where this single enzyme is able to break down the polymer cellulose into its constituent monomers, the sugars. And actually we're seeing even activity of the unmodified enzyme, but again, a solubility issue. So we improved the solubility of a substrate, and then we were actually introducing what we call solvent-based promiscuity towards the enzyme. And then we did temperature studies, so knowing that they're very thermally stable, we're able to show that enzyme activity has a new peak. So in aqueous conditions, the peak activity is around about 50 degrees Celsius, and then in the ionic liquid, we have a peak around 110 degrees Celsius. And this comes with a 30-fold enhancement of the activity. So not only have we introduced new activity for the enzyme by changing the solvent system, we're able to bring around a massive enhancement in that activity by being able to run it at higher temperatures. So this then showed us then that for this case, we were able to reduce the burden of enzymes from 3 to 1 for the processing of cellulose, but also do this at a much faster rate. So this was kind of a really exciting beginning, because in this case, we start to think of this as a platform technology. We're taking our enzyme, glucosidase, we were reacting it with a polymer in ionic liquids at temperatures above 70 degrees and releasing the monomer. And so if we're able to do this for biopolymers, perhaps we could start thinking of more unnatural substrates. And so the first thing that we've looked at then is the polymer polylactic acid. And this is used in plastics, either in coatings of coffee cups or often in those reusable plastic cups you see, the disposable ones. And so can we do the same thing? Well, we won't use glucosidase. Perhaps we'll take a step back and use an off-the-shelf hydrolase. This is one of the most common enzymes manufactured in the planet. This is used in a lot of those biological detergents. And so we're thinking then, so can we take an off-the-shelf enzyme, use some of those off-the-shelf ionic liquids and produce a new sort of demonstration of how we can use biocatalysis for recycling purposes. So can we then release the monomer from the polymeric substrate using an established enzyme system? Now, thinking of this temperature here, the reason I put this temperature here is this is above the glass transition for many of the plastics used that currently don't really have much of a recycling strategy, and polylactic acid being one of those. And so this should make things better as well. So this is work that Susanna's been doing. This is currently under review at Nature Communications. Fingers crossed that goes through. And so we always do a little stability study. Susanna showed quite nicely that we have, in this case, for this ionic liquid that was screened and chosen, we saw that there was an island of stability, thermal stability between 20 and 120 degrees Celsius. That means that we can truly use our enzyme at sort of some elevated temperatures. Long story short, it does what we wanted to do. So this is a picture of a post-consumer plastic. This is, I believe, a cup from Hotel Chocolat that's been cut into 100 milligram chunks. And Suzanne did a series of experiments, and this is just illustrating that in the ionic liquid alone we see some degradation, but once we have the enzyme present, we see significant degradation. And actually, within 24 hours, give or take, it's about 100% degradation and solubilization of the post-consumer plastic. And so this is what we see here. This is the mass loss of our chunks. And we're able to show that this was a demonstrative of the biochemical activity of the enzyme, and then knowing that we have mass loss within 24 hours, with some tweaking of the conditions optimization, we're able to show that we actually get full conversion to the monomer within 40 hours. So we get full degradation of the post-consumer plastic within 24 hours, and then we get full conversion to the monomer within 40 hours. And so this is kind of really exciting. So now we're able to show that we can actually, yes, we can do this. We can, using a off-the-shelf enzyme and with some off-the-shelf ionic liquids, through a modification strategy, we can start thinking of being able to open up new reactivities to quite efficiently degrade plastics, and start thinking of not only that, but degrading to their monomers. This could either be used as a feedstock for other chemicals, or could be used as a feedstock for repolymerization of plastic. And so we start thinking of introducing certainly a circular way of producing this bioplastic, which today currently doesn't exist. Great, so then if we can do this for polylactic acid, how about other scenarios? So the classic one everyone wants to look at is PET, so we've started looking at this as well. So taking a cutinase, this is in collaboration with a team at Rutgers University. This is the basis for all of those fancy petases that are in the news these days, except that this one is not being engineered, and we're seeing, can we basically start again? There's a lot of IP around those engineered petases, and so let's see whether we can go backwards and start again, using the fact that we can actually build in a different strategy, perhaps improve on that activity entirely. And so can we then, using ionic liquids, a off-the-shelf enzyme, with no prior engineering, and can we get this to work? Long story short, this is just preliminary work which needs working on, so Annika will be working on this from now on. And this is showing that we can get this to work. So we've shown on an unengineered enzyme, using our modification strategy, we are able to introduce reactivity of this enzyme towards the plastic to degrade it. Okay, in this case it's 7 days, but with a bit of tweaking, we will get this down to rapid. And so this, I'd say, in terms of what we're looking at is, I kind of refer to this as our destructive side. A lot of production isn't about destroying things, it's also about making things. And so the final example I wanted to talk about, don't need to know too much about this. This is just for any chemists who are interested in it. But this is a project we have with the Brazilian Biorenewables Laboratory, where we're looking at can we form drop-in biofuels. These are fuels which are directly convertible in engines, etc., for use from, in this case, waste foodstuffs to so we can convert waste foodstuffs to alkenes as a drop-in biofuel. And so we're focusing then on long-chain fatty acids, which will be part of eventually food wastes. Can we use this enzyme to form alkenes, which would be our drop-in biofuels? So this is different to the current batch of biofuels, which have fairly high oxygen content, and this high oxygen content means that they will eventually corrode engines if we have them too high in concentration. And so most biofuels have to be mixed with oil-based fuels anyway. And so this hopefully will be a route of circumventing that to develop methodology for creating fuels from a biofuel, which can be used in entirety for in engines. This is so catalyzed with the enzyme hydrogen peroxide. So we're thinking, well, why not use ionic liquids? Because this doesn't have much solubility in water. Its maximum solubility is around about 1 millimolar, which isn't very much at all. And so we're thinking, well, the ionic liquids are good solvents for really hard to deal with substrates, so maybe we can improve this. This is work that's being done by Jake, and this is currently in preparation. Again, skip to the end of this one. So stability studies, yes, again, same ionic liquid. Students seem to be fond of this ionic liquid. It seems to do everything they wanted to do. And stability is moderate compared to some of the others we're seeing, but anyone who's ever worked with a P450 will know this is pretty good, as this P450 in particular has a saturation temperature in water of about 45 degrees. So this is a pretty good enhancement of that. And the problem we're seeing, though, is that we can't use peroxide in the ionic liquid because it has a very short lifetime and it's actually incredibly reactive under this. But fortunately for us, and this is where we're now going to start exploring, I won't go too much into the details of this, but what we're seeing, this is the UV vis of the active group of the enzyme. I don't need to know too much about it, but what we were seeing in the presence of UV light, if we shine UV light onto this in the ionic liquid, we get characteristic shifts for a reduction of the heme which is our active site in this enzyme, to the active form in the absence of peroxide. So now we're going to use atmospheric oxygen and UV light to activate our enzyme such that it should work. And a lot of work is going into this to show that yes, indeed it does. And so this is just a GCMS trace of showing the consumption of the fatty acid over time after incubation with light and air and temperature in the ion liquid, and you see production of our alkene as a result. And so this is something that we're sort of currently preparing. Jake has since done a lot of temperature studies, a lot of other work, and we're moving forward with this one. So that's really exciting. So actually, we're now seeing that the sustainable solvent in our liquid is not only allowing for being able to operate sort of these reactions at greater efficiency. So I should have mentioned that whereas in water you can only run this reaction at 1 millimolar, we're looking at running it around about 28% now. So we can intensify this process and keep that same activity of the enzyme, and then this allows us to push forward in terms of generating more stuff faster, which is great. But particularly, what we're very interested and excited to look at is how we might use UV light instead of other oxidation agents as well.
What's the energy efficiency? I mean, how many energy in form of UV light do we have to invest? Is that negligible or is it?
Well, we'll come to that. That is a question I open up to the community. Is that's where we would love to go down the route in terms of understanding this more. So it's definitely going down the route of we want to understand more of the actual physics and the efficiencies, and look at those because of the economic cases on the cards. So skip ahead there for a second. But yes, this is so. In conclusion, I've shown that sort of in the group we have a methodology for basically bringing biology, kicking and screaming out of water, its comfort zone, into an environment which is more amenable for industrial transformations. So we can keep the structure, the activity, and actually, if we choose our solvent correctly and our temperature range regime correctly, we will get an intensification and enhancement of that analysis, which should hopefully be very useful. So, which moves on to, I was asked to think about those challenges and opportunities for net zero, and as I started with this, I started thinking about the different aspects of this work. So this is, I like to think of this work as being a platform technology, so we are looking for other reactions that we can do. If anyone has any use for stabilized enzymes that can operate at up to 150 degrees Celsius in terms of biosensing for whatever, or for other things, some other ongoing projects we have I haven't shown, and looking at sort of using redox enzymes, so we use this solvent to strip out all of the requirements, the cellular requirements, and replacing it with some sustainable and very easy to control electrochemistry. Or we have some dormant projects, I've actually just submitted a nucleus LC grant for looking at carbon capture, so the same system should be able to stabilize enzymes which can convert CO2 to carbonate very efficiently. And then thinking on the other side of things, we have we can use these. There's another dormant project at the moment is thinking of other aspects of net zero, where we can make gains in decarbonisation, reducing the energy requirement is in therapeutic stabilization. So a project where we've looked at, we can actually generate materials where we can envision a distribution system for life-saving therapeutics such as vaccines that doesn't require the cold chain. So we can stabilize them against temperature, so therefore we can have a route to having biotherapeutics which don't require freezing or refrigeration, which not only democratizes the access to those therapeutics, but it also reduces the energy and therefore the carbon footprint of those materials as well. Those things are, as I said, they're dormant, so we can help and things out there for people to get involved. This is the Universal Symbol for Engineering, and so what we're looking for also is the determining actually exactly those things. So things like the opportunities here is looking at the lifetime assessments, those techno-economic analyses, the efficiencies. Do they, I can say as much as I want to chemists, but yes, it's using lights. Yeah, right. We can recycle this over and over again. Absolutely fantastic. But I haven't got the money with my mouth just yet. So there's opportunities there. And so I started thinking this is where this kind of starts turning into a manifesto at this point. Is where else then in terms of opposition? So these low TRL sort of technologies and how can a net zero center sort of help us move forward, is sort of thinking about the actual application of deployment side. So we have an idea of where we want to go, but we don't really have that technical knowledge or sort of feedback from industries. This is something that I think would be nice to see from help from colleagues or sort of the opportunities here in engaging the industry, particularly for commercialization and adoption of these products. So at the moment, the brief conversations I've had with industries have been around that. Oh, yeah, that sounds great. Have you shown this reaction? This very specific reaction that, well, not yet. I mean, give us the money we will, and then we'll figure something out. So we need that kind of the opportunities there in sort of getting sort of facilitating commercialization and adoption and sort of talking to potential industry partners. Then, as I said, this is where the manifesto part comes into it. I was thinking, how then do we at King's make the best of our environment to facilitate all of this? And I think when I've got compared to see some of the other universities in London, we have an excellent policy institute. So something that I think a big benefit of at King's is to sort of interface with some of the mechanisms that already exist in the college to interface them with government as well. So I think as scientists and engineers, we should really be thinking about if we're going to complete the circle on getting our technologies through to deployment, is to engage in the policy writing and actually policy validation, which will then require speaking to both the public, politicians. At the moment, public are largely on our side. We're seeing certain stripes of politicians are largely not on our side anymore, and so we need to re-engage in both politicians and public interest. Why? Well, because if we engage on this side, sort of engage with the public and policy validation, we can start thinking of regulation. And this isn't regulation to stop the uptake of net zero technologies or sustainable technologies, it's to ease the pathway to allow for sort of both public and politician trust. And the greater the trust you have that everything's going to work well, the greater the uptake and the greater that this will facilitate this side of things. This side of things is pretty easy. There's a reason for that. It's got cash, and I wouldn't quite like to chest and then we can really get things going. So that's how we complete the circle, hopefully, to the net zero center. So, with that, I'd like to thank my group for the wonderful work they do. So, present and past. This is an old photo. Collaborators over the years and funding for everything that we do. Thank you for your attention.
Thank you very much for the presentation and for the spin on the net zero. So thank you very much for that. So before I say anything else, I just want to see if there is any questions from the group. Yeah?
I have a quick question on the plastic recycling. You say on the liquid and the enzyme. So from your graph, I'm not sure if I understood it correctly, but it seems that most of the efficiency from the recycling comes from the ionic liquid,
Yeah.
and then the enzyme adds on top of it.
Yeah, and so actually what I didn't show here was that if you leave it for long enough with just ionic liquid, it'll go back into itself.
Oh, okay.
And so what we're seeing here, this allows us to produce and get to 100% yield of our monomer, and then extract it before it starts, but otherwise it never goes to completion in ionic liquid.
Okay. Any questions?
Yep.
Thank you.
I mean, we need help on the extraction side of things, so any engineers out there who can help us on that process development, that is absolutely critical, but yeah, it's there, so a bit more about that.
Yeah, just had a question as well on this reaction. Sorry, I come from a power system point
Yeah,
of view,
no,
so I don't really, maybe I don't understand. When you explain the speed of the reaction, I think you said some experiments took 24 hours,
Mm-hmm.
and then you mentioned some other reactions were a bit slower, maybe a few days. So is that important that it's fast for plastic to degrade? Because I mean, when I think about it, I wouldn't really worry if it takes a few days rather than a few hours.
It's a good question. And again, this is where maybe some of the assessments come into things of like, what is the advantage? Because at the moment we're seeing that sort of, and my students are doing a very good job of this, of optimizing the system so to do it as fast as possible. But do we need that? I actually don't know, and that's where kind of the linking with people.
Because
help.
maybe, you know, if you can make the process, I don't know, maybe less, you know, more efficient and less energy demanding, maybe, you know, you could, I don't know, maybe you could sacrifice some of the time and, you know.
Absolutely, and we're seeing that with the, it's actually for the stabilization of the biotherapeutics, what we're seeing there is actually, currently we've got overkill in that we've stabilized, so in that particular paper we were able to stabilize the therapeutic up to 140 degrees Celsius, which meant that it had a near infinite shelf life. But that came with a loss of some of the bioavailability, and so there's obviously a balance there that we need to do, but this is a dormant project now, sadly. But this is there is a balance there, definitely, in our process.
I think your point on the Policy Institute was very interesting. So, do you know anyone there that we could engage in the Net Zero Center?
Yeah, I mean, I've met with, I don't know if they're still there, but Mark Kleinman was very interested in sort of engaging more in science. And actually, before the pandemic, was tentatively trying to go and build some links, but that kind of died because of the pandemic, along with lots of other things.
So maybe we can invite them for another seminar?
Yeah, no, absolutely. I think it's definitely worth reaching out. We have links, I think, yes, particularly with the other sort of vague centres that are correlating as well. The NERC, discipline hopping, the people. I'm blanking on the person who's involved, but he's also involved in that and the Policy Institute. So we know definitely links that we can make build there.
One last question.
Yeah, sure.
Sorry.
Thank you.
Because in your ionic liquids, you mostly take things apart.
Yeah.
Recycling. For example, we do a lot of inorganic synthesis in ionic liquids as well. Synthesis.
Okay.
Do you think your enzyme ionic liquid mix would help to maybe accelerate this?
Yeah, I mean, it depends on the reaction, but provided we can find the enzyme to match the reaction that you want to do, and actually in many cases, just a similar enough reaction, if we just tune the ionic liquid to be amenable for that reaction, then I see no reason why we couldn't do, provided it's a reaction that we can find an enzyme for. But, yeah.
I think there are some of your students here as well in the room.
Yeah.
Some of the people that maybe did the work, some of the work you presented today, right? So are this group?
Yeah.
So thank you for being here and for your contribution.
Yeah, absolutely.
I think
Yeah.
that's it.
Well, thank you very much.